gentlemen, let me present you the online assisted remote practical session in conducting biology experiments. So I am Prasad here. The online practicals were the only available method during the COVID-19 pandemic we all know about, and most practicals conducted online consisted of demonstrations only. So there were no engagement of the students. The students were looking at the practicals that were conducted by the demonstrators. So the reflective analysis indicated that we need to modify this. We need to engage the students, and therefore we developed the method called the online assisted remote practical sessions. So the conventionally we all know that the biology or engineering or whatever, the practicals are conducted in the lab or the laboratory environment or conducted in the designated field, and they cannot be completely replaced. But we are redesigning a selected set of practicals, not all experiments, to be conducted from home environment with the online assistance. So the difference here from the rest of other modes is, so the staff need to select the practicals which are suitable to conduct in the remote locations. Students are required to do some preparations for this practical session by collecting the materials and organizing those things, and sometimes even developing some instruments. And then it made it into a very important learning step, the active learning step. Though we say that OARP is a new method, OUUK, and this is again the website a screenshot from the Sri Lanka Association for Advancement of Science, where they made available some kits. However, there is outstanding feature in this here, what I am presenting you, because this contains the live demonstrations, teaching and supervision, and the engagement of the students in these experiments. The week 1 was conducted in the online assisted remote practical session. What I am presenting here is the updated one which we are practicing now. However, earlier during the COVID pandemic, we could not conduct the session 2, 3, 4 or 5. They were conducted as a demonstrations. We are conducting the session 1 as the OARP. We are maintaining the same momentum, and we are calling the students to the lab and conduct the rest of the sessions. So we are giving students with this experience of conducting practicals under the remote supervision on one hand side, and also the conducting in-lab practicals. So the workflow of the OARP session was like that. We started at 8:30, and there was an introduction conducted, and then the practical demonstration was conducted. And after that, we assigned them into the breakout room, and we checked whether all materials are there and flowcharts are drawn. Both flowcharts are mandatory because that's indicate their preparation. And we had a discussions after noon and evening. To devise this ORP method, in fact, we had to have a lot of work to do. So we have to identify the practicals which can be done at home, and not only that, we couldn't supply any material from our labs. We have to suggest some alternatives for even for the chemicals, even for the materials, the material in their home. Now we will see how we have done it in the lab. So this is the first online assisted remote practical session we are doing for this. So I do hope that all of you are in front of your computers or the phones. You may be in a different setups. Most of you would be at your residence. However, mind you, now you have to change your mind as if you are in the lab. So what I would do today is, first of all, I would be giving a brief introduction. Then I will hand it over to the team of staff members here. So they would be demonstrating you the practicals that you are doing today. And after that, you would be getting time to show your preparation. So your small lab that you have set up in the home. And for that, you would be put into the different rooms in the Zoom, and our staff members will be looking into your preparations in terms of your materials, your flow chart, and we will also be checking your attendance, and after that you have time to do your practice, and then we will be calling you at different time intervals to see your progress. We have the final discussion at 3:30, so you are supposed to be online, and if there are difficulties you can come on and off during while you are doing practicals. However, you should be online in the designated time. Online assisted remote practical session is a new way of doing practicals, and this has many positive sides. You as a students are preparing yourself for practicals. With the material at your disposal, you are now doing the practical on your own. You are being monitored by the staff members remotely. And when we are asking questions, there are two domains we are checking. The first domain is the knowledge based learning, the other one is the skill based learning, right? You need to record everything. Whenever you are doing, take photograph of your materials, photograph of your observations. We are asking you to submit a report on your online assisted remote practical, right? So you know that that is going to be assessed, getting an experience of working in a virtual group. So this is the real working environment nowadays that is existing. So now the boundaries of the countries, the nations, and even the geographical barriers have disappeared because of this virtual environment. You know that people who are working in a projects, they work in a different countries, they interact through this virtual environment, and today you are doing the same thing. So you can talk each other, doesn't matter, through this online, right? Whenever there is a question you can ask, even you can call a name and ask as if you are in a lab. I will hand it over this session now to Rochana and his team.
This is about experiment number 1. This one is about the effect of chemical substances on the membrane permeability of the different types of plant materials. This, and then I'm going to use acetic acid. You can use vinegar, ethanol, and then I'm going to use 50% ethanol. For that, you can use surgical spray instead of the ethanol. So after you, first you need to take the 5 different glasswares. In here, I am going to use 5 different beakers. You can use test tubes or any other.
Now we are going to focus on the practical number 5, the determination of the water potential of the plant tissue. For that, you should have potato strips and sucrose solutions, and then first you have to make a sucrose solutions. For that, you can use 1 ml of solution, or you can use the table sugar instead of the sucrose solution. You know the molecular weight of the sucrose. Molecular weight.
To observe the effect of auxin on leaf abscission, we have to first remove the leaf coverage. So if we can take 2 separate. Then we have to apply, for one petiole, we have to apply the lanolin, and for the other petiole, we have to apply auxin.
And also someone can use chalk or calcium carbonate to provide carbon dioxide, and also you can use calcium hydroxide. Calcium hydroxide means we normally call aluminum. We can part of the plant like this. Then we have to add this plant into a solution, prepared solution.
So we are going to start the experiment number 11. It's based on the heat effect on enzyme activity. So for that, you need to have soap moon seeds and a mortar and pestle, and you should have starch solution. For that, you can use unprocessed denatured. So to prevent that, keep your mortar and pestle in the fridge for some time and get it cool, and take a few moon seeds into the remote and
We are going to do experiment number 28, the effect of ethylene on ripening of fruits. For this experiment, we need unripe lemons and also ripe banana and unripe banana. So for one, we need another 2 Ziploc bags. So for one bag, I have labeled it as A. Here I have put unripe 2 lemons, and for this bag, I am going to put this unripe banana. This one, the 2 lemons, they have ripened well.
students, next we will start experiment number 9, setting up the hydroponic culture. Right? So, I think you can see this one. First, you want to have a pot with holes and some plants. So here we have set up plants, and I will show you one. So first you can insert plant like this, and we are taking coir dust as supporting material. So you can.
Students, we are preparing after doing the last experiment. We are assigning you to the breakout rooms. So there you have to show your materials and the flow chart. So get ready with those until we start the next experiment.
How they have done it at home. So this is one of the experiments. So I'm not going to elaborate this experiment. These are biology experiments. So you would see that. So instead of using the glass, usually we are using the test tubes. But here they have used what they had at their home, and you can see the different colors have come because they have put different acid, base, and other alcohol types solutions here, and that's why they are giving the different colors. Experiment where they do the water relations of cells and tissues, and this you can see they have used some of, and they have somehow made the series dilutions of solutions. So they explain us how they did it. So they have achieved the learning outcome that is stipulated. And this is another one. So you would see this is called the pottometer, and this we use for the measuring the transpiration in the plant. You can see that how they have done it at their home. So somehow they found a method using the syringe and the saline tubes, and they measured, and they have shown lively to us how they were doing it. And then you would see they have even set up the hydroponic setups at home. And this is one of the very good practical applications that they can even maintain throughout at their home. It was very much evident, and they were identifying the alternative ways to conduct experiments, and there was a learner autonomy, and we could see even the creativity and innovation in them. And the learners' engagement in inquiry learning was also apparent, and we could see that they were maintaining the lab corners at home, which is a novel experience to them. And there were drawbacks because this was not a perfect method. So the drawbacks that they have shown was the connectivity issues in the online sessions, and also the expensive nature of some materials. In fact, actually we asked them to use the very cheap materials, and the unconducive home environment. Obviously we know that not all homes are having the peaceful situations. And the other one is the lack of peer support and teamwork at the university. That is what they have mentioned. So let's have a look on the survey that we carried out. So what we did was actually we did the effect of this practical method using both qualitative and the quantitative way. So here you would see we have asked several questions, and you can see 169 responses were there. In addition to that, there are some interviews as well that we conducted. So one question was, what are the most preferable features in online assisted remote practical compared to the conventional lab practicals? And you would see that it says it allows to work at my own pace, which is a very important thing in the open and distance learning, and it offers the freedom of doing experiments, and it increases the creativity, and it entrusted me responsibility, and also it made me understand the experiments as they are doing it by themselves. And then the next question was, according to your view, what are the two major disadvantages in online assisted remote practicals? And you would see that lack of live engagement they have felt, though they were in the remote environment, lively connected. And the other one is the most of the time, that most highest thing that they mentioned was the time taken to organize the material. So of course it takes some times for the material. And the second one is the cost that they have mentioned. And you would see that there are two. So the online assisted remote practical session of BYU 4300 is the first ever such experience I received in the USL. You can see, I mean, the 1 and 2 are the positive results, and 3 is the intermediate, and 4, 5 are the negative responses to that. So you would see that here, so the most of the students have responded positively. Nearly more than 70% say that it is their first such experience. And this is the online assisted remote practicals were more beneficial than on-site practicals. You would see there is a mixed, somewhat mixed responses. However, the other one is the online assisted remote practicals provided me good opportunity to carry out experiments in my own than working in groups. So actually many students have agreed that yes, training I received through the online assisted remote practicals will help me to carry out in-lab practicals effectively in subsequent sessions. Yes, of course, because school, this is a very good question. As a school teacher, because there are so many school teachers in the batch, it became useful to try out similar approaches. Idea how they could develop the background to do this type of experiments even at the school curriculum level. So the online assisted remote practical using alternative methods prompted me to set up a lab cone at my home and conduct some experiment that I am interested in. Yes, you can see this is a very good habit that we are developing. So they are now extending it in such a manner, maintaining it as a lab cone as well. And I enjoyed online assisted practicals more than conventional on-site practical because it conferred me a lot of confidence in carrying out experiments. So you would see there's a very high, again the positive trend is highlighted over that I achieved the same expected learning outcome even though online assisted practical session. You can see that they feel positive responses to this one. When we introduce this, there are a lot of objections coming up. Even there were complaint went to the hierarchy of the university, but still we moved in. And yeah, so I think there are nearly 60 students today, and you had a somewhat different experience because you worked remotely by doing an experiments, and you were connected time to time with the main session, and you were given the facilitation or the assistance. So we would like to know your experience a little bit from your side. You can honestly say what is your experience, because we really want to know about it. That's why I convened this session. I have been convening this same session, like this throughout all 3 days, past 3 days, to know your perceptions about what you did today. And this this session is different due to its nature and might be having some different views, comments, experiences.
ඇතට මේක ගොඩක් හොඳ එක්ස්පීරියන්ස් එකක් වුණා. ඇත්තටම කිව්වත් අර කියන්නේ මැටීරියල්ස් කලෙක්ට් කරන තැනේ ඉඳන්ම චැලෙන්ජින් එක කියන්නේ දැන් ගෙදරට කිව්වම මෙච්චර ගොඩක් අර ඒ කියන්නේ බඩු ගේන්න කිව්වම දැන් ගැන එක්ස්ප්ලেইন කරලා මේ zoom එක තියන කලින් බඩු ලිස්ට් එක දැක්කම බය වුණා. ඒ කියන්නේ ඒක තියෙනව නිකන් අර හිතුවේ ගොඩක් සල්ලි දීලා ගන්න ඕන කියලා. බට් ඊට පස්සේ සර් එක්ස්ප්ලেইন කරාට පස්සේ ඒ බඩු ටික හොයාගන්න එක එක අර හයිඩ්‍රිලයි අර කැල්සියම් හයිඩ්‍රොක්සයිඩ් හොයන අර ඒ වලටත් අර ඒව හොයන විදිහ ඒවා එක එකනට කියලා ගෙන්න ගත්තා. අර එහෙම ඒව ඔක්කොම අර ගොඩක් හොඳ එක්ස්පීරියන්ස් එකක් ඇත්තටම. බඩු කලෙක්ට් කරන එක ලිස්ට් එක හදාගෙන එක ටික් කරලා ගෙනාවද නැද්ද කියලා බලලා අර එකට කලින් දවසේ රෙඩි වුණේ ඒ හැම එකක්ම ස්ටෙප් එකක්ම අලුත් එක්ස්පීරියන්ස් එකක්. ඇත්තටම අද ගොඩක් අර අපි අර ලේසි නෑ ඇත්තටම අර තනියම කරන එක. බට් අර ඇත්තටම මේ සතුටුයි මෙහෙම කියන්නේ යමක් කරගන්න පුළුවන් කියන හැඟීම දැනුණා මේක කරාම. ඇත්තටම අර ගොඩක් හොඳ එක්ස්පීරියන්ස් එකක්. සර්ටයි හැමෝටම ස්තුතිවන්ත වෙනවා. ඒ කියන්නේ මේ
තව එවැනි අදහස් තියෙනවද?
Okay. So the how we so it was very independent and it was very interesting to start off the practical by bringing the gathering all the items that is needed by ourselves. And I also felt like there was a bit of struggle since maybe that is because I had to work alone. So I also had like a very intense morning time when I compared that with evening. I just felt like that. Yeah, but it was very interesting working alone at Mini Lab. Thank you for providing the opportunity.
Today it's been from this practical we learned that how to do a practical within a available thing, right? Without now we don't have a proper chemical with us. Like we use instead of HCl we use citric acid, and instead of NH4H like the base items we use soap water. Now we know how to use available things for our purposes. So that is very useful. We are just thinking in a practical way. This is very nice, and now we have made a photometer, and that was a good experience because we are just try to make some of the tools that related to do our practical completely very good man. So that was a big experience I get. And that Albert solution also we can use that in our real life to make some of the trees to grow like strawberry plantations, like so such a good practical. I felt very interesting this one, and I just think that plant physiology will be very hard, and this one is an online practical session. How can I do like there are so many doubts I have in the beginning, but now I feel good. It was good, and this one is very better, sir. So
Oh,
සර්
ලක්ෂ්මන් ලක්ෂාන්
සර් මට කියන්න තියෙන්නේ මේ අපි උදේ හාෆ් එකේ අතම කරද්දි එක්ස්පෙරිමන්ට්ස් බ්‍රේක්අවුට් රූම්ස් වල කරේ. එතකොට මේ අපි ගොඩක් අර ළඟින් ඒ කියන්නේ ඒක ගොඩක් අපි ඒක දැනුණ විදිහටම කරා. But when we are in the main room and when we are doing experiments, the concentration comes and goes, and I think it's better with the breakout rooms. So in 10 to 12 students in there, so demonstrators are asking questions every time, so we have to very focus. So I think it's better to do all the experiments in the breakout room. That's my idea.
Yes, sir, but to say the truth, initially it was a headache before starting the experiment. It was kind of dark තියෙනවනේ හොයාගන්න ඒ වගේ expression එකක් තමයි we had, but when you are doing this, it was the best experience I had in my life. And now the initial expression has totally changed.
what would you do differently if you are going to do this practical again?
Yes, sir, actually getting the demonstration and the discussion from the same demonstrator would be more beneficial, because when we come to the discussion, then that would be that we felt somewhere areas were not accordingly, that may in some areas I think we can get a clear picture.
අවුස ඇත්තටම වැදගත්ම දේ තමයි ෆ්ලෝ චාර්ට්ස් අඳින එක නතර කරන්න එපා. ඒක තමයි ලොකුම අපිට වැදගත්ම වෙච්ච දේ. ෆ්ලෝ චාර්ට් එක අඳින එක ඇත්තටම ඒක කම්පෙල් කරා. අනිවාර්යයෙන්ම කරන්න කිව්වා. ඒක මැන්ඩටරි වුණා. ඒක කරන්න කියපු හින්දා ඒකත් එක්ක එදිච්ච හින්දා තමයි ඇත්තටම ප්‍රැක්ටිකල් එකට එන්න කලින්ම ඒ ගැන නොලෙජ් එකක් පොඩි දැනීමක් ආවේ. පොඩි නෙමෙයි ලොකු දැනීමක් ආවේ. ඒක නවත්තන්නේ ඒක නම් කොහොමද නවත්තන්න එපා කියලා තමයි කියන්න තියෙන්නේ. ෆ්ලෝ chart එක ගොඩක්ම ඇත්තටම දැගත් උනා.
හොඳයි.
කරගෙන යද්දී ප්‍රැක්ටිකල් එකට එද්දිත් ඒතරම් කන්ෆියුස් වෙලා නැහැ. ප්‍රැක්ටිකල් එකට එද්දී යම් කිසි දැනීමක් තියෙනවා ප්‍රැක්ටිකල් ක්ලාස් සෙෂන් එකට එද්දී. අපි කරන්න යන්නේ මේක කරන්නේ මේක. ඒ වගේම තමයි අර අතර මගදී කරන පාර්ට් එකේ උනත් අර කීපයක් එක දිගට අපිට කරන්න ඕන. ඒ කියන්නේ පොඩි රෂ් එකක්, ලිටල් බිට් රෂ් එකක් තිබ්බා. එතකොට ඒ රෂ් එක අතරතුරේදීත් අර ෆ්ලෝ chart එක තිබ්බ හින්දා අපිට පුළුවන් වුණා ඒ ප්‍රොසීජර් එක ගන්න ෆලෝ කරන්න.
කියන්න කැමති කෙනෙක් මේක ඉස්සරහට පවත්වාගත යුතුද මේ විදිහට ෆස්ට් සෙෂන් එක මෙහෙම කරලා ඊටු සෙෂන්ස් ලැබ් එකට ගෙනල්ලා කරන විදිහක්ද නැත්නම් ඔක්කොම සෙෂන්ස් අපි ලැබ් එකේ කරන එකද හොඳ?
This is better, sir, I think.
This one is good, sir, because every student will get experience that we can do any practical in our home also. So this one is good, sir, for the future students also apply this method. This one is very good.
Okay.
Yes, sir, this method is really good. Because
මෙහෙම කරන්න පුළුවන් නම් වටිනවා. ඒ කියන්නේ අර ඇත්තට මේක කරගන්න පුළුවන් කියලා දැන් දැනෙන නිසා ඒ කියන්නේ ගොඩක් අට ප්‍රයෝජනවත් ඒර දුර ඉඳන් එන අයට එහෙම උනත් අර තව සෙක්ෂන් එකක් අර ඔන්ලයින්ම කරොත් එහෙම අර එහෙම කරත් හොඳයි කියලා මට හිතෙනවා. ඒ කියන්නේ අර මේක සක්සස් කියලා දැනෙන නිසා.
ඕකේ. ඔව්. තව තියෙනවද අදහස්?
ඇත්තටම මේ ක්‍රමය ඇත්තටම හොඳයි සර්. මේ මොකද මමත් සර් දැන් ඒ ලෙවල් ඉඳන් අපි සර් මේ ප්‍රැක්ටිකල් කරනවානේ. දැන් ඒ ලෙවල් අපි කරන්නේ සර් ගෲප් වයිස් කරන්නේ. ඉතින් ඒකේ සර් අපිට මේ ඇත්තටම ප්‍රැක්ටිකල් එක පිළිබඳව අවබෝධයක් ගන්න ටිකක් අමාරුයි සර්. මම හිතන්නේ මේක මම කරපු හොඳම ප්‍රැක්ටිකල් එකක් මේ ඒ කියන්නේ මට හැම දෙයක් පිළිබඳව සර් මේ කරපු ප්‍රැක්ටිකල් අටේම ලොකු අවබෝධයක් ආවා සර්. ඒ වගේම මේ දැන් අර අපි ඒ ගන්න මේ සොලියුෂන් එකට මේ මොනවද පාවිච්චි කරන්න පුළුවන් අනිත් දේවල් කියලාත් මම මේ පාර ප්‍රැක්ටිකල් එකේ මම ලොකු අවබෝධයක් ගත්තා. ඉතින් මේ ක්‍රමය ටිකක් හොඳයි. ඒ කියන්නේ ටිකක් හොඳටම හොඳයි. මේකට පුළුවන් නම් කන්ටිනියු කරන්න කියලා මම කියනවා.
Actually, we were asked to find some 5 test tubes like that for this one, but during the experiments we couldn't discard the setups. So that was a big problem we faced during this time, because we have to find so many glasses and all the test tubes we brought. Actually, we only 5. Other than that, all other experiments we had to find 20 to 30 glasses for that.
The responses have changed. You can see, okay, now we will look into the conclusion. So the conclusions are the learners successfully took part in biology scientific experiment at home with live instructional support through this online assisted remote practicals, and this can be extended even after the pandemic with careful review of reducing the drawbacks. Yes, we have done it, and we have now introduced it as a standard practice in the Plant Physiology course, which is a second year course of the BSc degree program. So thank you very much.